See Part 2, Muscle Contraction for the AAQ Medical Science Unit 1 exam. So we'll be looking at, we did this on the last video, this one would be Principles of Sliding Filament Theory of Contraction of Skeletal Muscle with the roles of actin, myosin, troponin, tropomyosin and ATP. The different properties of slow and fast twitch muscle fibres, calculations involving proportions of slow and fast twitch fibres and in change of length of sarcomeres and muscles or of change in muscle mass, neuromuscular junctions and roles of neurotransmitters, antagonistic muscles, the role of tendons in attaching muscles to bones, origin and insertion, action of antagonistic muscles as illustrated by flexing and extending the forearm. So an overview of muscle contraction. The myosin heads slide actin along the myosin, causing the sarcomere to contract. Simultaneous contraction of many sarcomeres cause myofibrils and muscle fibers to contract. When the sarcomeres contract or shorten, the H zone gets shorter, the I band gets shorter, the A band stays the same, and the Z lines get closer. So the sliding filament hypothesis. So the thing that triggers the whole sliding filament muscle contraction is, first of all, an action potential or depolarization will travel down the T-tubules of the sarcolemma, and that will trigger the sarcoplasmic reticulum to release calcium ions. And these calcium ions cause tropomyosin to move away from the actin binding site. So if we look at the diagram here, the blue is representing actin, the reddy pink is representing myosin, and then the green is representing tropomyosin. Now, we can't see the diagram before this, but you would have another diagram where the green tropomyosin is actually blocking the binding site on the actin. So the tropomyosin, the green, would be here blocking the actin binding site. But when calcium binds to tropomyosin, the tropomyosin moves out of the way and reveals the actin binding site, making them available for myosin to attach. Then the myosin heads attach to the binding site on the actin, forming an actin-myosin cross-bridge. And then the myosin head forms a power stroke, so it slides the actin over the myosin. The head tilts and called a power stroke. It tilts and pulls the actin over it. And at this point, ADP and PI is released from the head. Then an ATP molecule binds to the myosin head, and this causes the myosin head to detach from the binding site on the actin. And it also activates ATP hydrolase, which causes the ATP to hydrolyze into ADP and PI. And the energy released from the ATP causes the myosin head to tilt back to its original position. And then the myosin head will reattach to a new actin binding site further along the actin, and the whole cycle will happen again. So first of all, calcium binds to tropomyosin. Tropomyosin moves out of the way to reveal the binding site. Then the myosin head will attach to the binding site on the actin, forming an actin-myosin cross-bridge. ADP and PI get released, and the myosin head tilts, does its power stroke, tilts forward, pulls the actin over it. Then ATP attaches to the myosin head, which causes the myosin head to detach from the actin. Then ATP hydrolyzes ATP into ADP and PI, and the energy released is used for the myosin head to tilt back to its original position. And then it binds to a new site on the actin further along, and this just continues until it stops contracting and the calcium ions go back. So here we've got the role of actin A, myosin M, calcium ion C, tropomyosin T, and ATP in muscle contraction. So the first thing that happens is depolarization spreads down the sarcolemma via the T tubules, which causes calcium to be released from the sarcoplasmic reticulum, which diffuses into the myofibrils. The calcium ions then bind to the tropomyosin, causing it to move and exposing the binding sites on the actin. This allows the myosin head with the ADP attached to bind to binding sites on the actin, forming an actin-myosin crossbridge. The myosin head changes angle, pulling the actin along, which is known as the power stroke, using energy, and ADP is released. Then a new ATP binds to the myosin head, causing it to detach from the binding site. The hydrolysis of ATP by ATP hydrolase releases, which is activated by calcium, releases energy for the myosin head to return to its original position, and then the myosin reattaches to a different site further along the actin, and then this process is repeated as long as this calcium ion is present. But then after contraction, during relaxation, the calcium ions get actively transported back into the endopolar reticulum. You're also using energy from ATP. Tropomyosin then moves back to block the myosin binding site on the actin again, so the no actin myosin crossbridge is formed. And this is to make sure that you don't continuously contract your muscles. Then looking at the difference between slow twitch and fast twitch muscle fibers. Slow twitch are specialised for slow, sustained contractions like posture or long distance running. They produce more ATP slowly from aerobic respiration, and they fatigue slowly. They have a high proportion in the muscles, and they're used for posture. And so posture muscles like the back and the calves. Also, the legs of long distance runners will have a lot of slow twitch. The structure, this is where a lot of people lose marks. Structure is slow twitch. They have a high concentration of myoglobin, which stores oxygen for aerobic respiration. Many mitochondria for the high rate of aerobic respiration. Many capillaries to supply a high concentration of oxygen or glucose for aerobic respiration and to prevent buildup of lactic acid, causing muscle fatigue. Then you've got fast twitch, which is specialised for brief, intensive contractions like sprinting, produces less ATP rapidly from anaerobic respiration, fatigues quickly due to high lactate concentration, has a high proportion in the muscles used for fast movement like biceps and eyelids, a lot in the legs of sprinters or weightlifters. There's low levels of myoglobin, lots of glycogen, which can be hydrolyzed to provide glucose for glycolysis, anaerobic respiration, which is inefficient, so there's large quantities of glucose required, high concentration of enzymes involved in anaerobic respiration in the cytoplasm, and there's a store of phosphocreatine. So now looking at calculating percentages of muscle fibers. If an athlete has 60% slow twitch muscle fibers, what percentage of fast twitch do they have? We just do 100 minus 60 to get 40%. Calculating the change in sarcomere length during contraction. A sarcomere in a relaxed muscle is 2.5 micrometers long. During contraction, it shortens to 1.8 micrometers. Calculate the change in length of sarcomere, the percentage decrease in sarcomere length. Well, the change in length is just initial minus final, so it would be 2.5 minus 1.8 to get 0.7. And then to find the percentage decrease, you do change in length over initial times by 100. So you would do 0.7 is the change, divided by the original is 2.5. So 0.7 over 2.5, times it by 100, you get 28%. Percentage increase in muscle mass. A person begins a strength training program. Their initial muscle mass is 28 kilograms. After 12 weeks, their muscle mass increases to 30.5. Calculate the absolute change in muscle mass and the percentage increase in muscle mass. So the change in mass is final minus initial. So 30.5 minus 28 is 2.5 kilograms. That's our change. And then to find the percentage increase in muscle mass, you do change in mass over initial. So 2.5 divided by initial, change is 2.5 divided by initial is 228, times by 100, and you get 8.93%. And then looking at the neuromuscular junction. So a neuromuscular junction is very similar to a synapse, but instead of there being a post synaptic neuron, you have instead the muscle sarcolemma. So this is a motor neuron. When an action potential reaches the motor neuron, it'll cause calcium ions to open, channels to open, calcium goes in, and the vesicles will move towards and fuse with the presynaptic membrane of the presynaptic neuron, and they'll release acetylcholine neurotransmitter into the synaptic cleft. Then the acetylcholine will diffuse across the cleft and it will bind to receptors on the complementary receptors on the sarcolemma. And that causes depolarization of the sarcolemma, which then travels down the T-tubules, which triggers calcium to be released from the sarcoplasmic reticulum, and then the whole sliding filament hypothesis starts. So the roles of the components: acetylcholine is a neurotransmitter that initiates muscle contraction. Calcium ions trigger release of acetylcholine from the vesicles. They make the vesicles move towards and fuse with the synapse, the presynaptic membrane. The sodium ions enter muscle fibers to start depolarization. Acetylcholine esterase is an enzyme that breaks down acetylcholine to stop the signal so we don't continuously have our muscles contracting. And then finally, antagonistic muscles work in pairs. When one contracts, the other relaxes. For example, when the biceps contract, the triceps relaxes, etc. The tendons connect muscles to bones and they transmit the force to move the skeleton. The origin is the fixed attachment point of a muscle to the bone. The insertion is the movable attachment point, which is pulled during contraction. As an example of antagonistic muscles, forearm movement. When you flex, the biceps contract and the triceps relax and the lower arm moves up. When you extend, the tricep contracts and the biceps relax and the lower arm moves back down.